In this video, we want to look at the mechanisms of activation of smooth muscle. We're going to find that it differs somewhat dramatically from that of skeletal muscle or cardiac muscle. Now, it's important to note that smooth muscle is found lining many of the hollow structures of the body, excluding the heart, of course. And that would include the respiratory tract, the GI tract, the urogenital tract. It's actually associated with some skin structures. And finally, as part of the intrinsic muscles of the eye, controlling the size of the pupil in the iris, as well as regulating the shape of the lens for focusing of light. Now, activation of smooth muscle or inhibition, there's actually a possibility of either, can be mediated neurally through autonomic innervation. We have autonomic neurons at the top left here, or via capillary delivery of hormones. So endocrine control also exists. Regardless of the delivery mechanism of that chemical signal, we're going to find that they bind to receptors found in the membrane that are sort of clumped together in regions. And this is allowed by structures called calveoli. And calveoli are lipid rafts that they're high density regions of membrane that help keep membrane associated proteins lumped in one spot. Right? So it sort of collects all these associated proteins in one region, allowing for coordination of smooth muscle control. Now, what we find is that they come in quite a variety here, right? In the middle here, we have a G protein coupled receptor. In this case, that's linked to opening of a calcium channel, allowing calcium influx. We're going to find that a rise in intracellular calcium is always our stimulus for muscle contraction. Doesn't matter which type, smooth, cardiac, or skeletal. On the right here, we have our ligand binding to a receptor that is metabotropic. Metabotropic receptors are linked to the production of second messenger molecules, shown here by the little triangle. And in this case, that little second messenger molecule is going to bind to a receptor and open a channel in the sarcoplasmic reticulum membrane. In this case, allowing the stored calcium to diffuse outward into the cytoplasm, again, adding to that rise in intracellular calcium that's going to be our stimulatory factor. Finally, on the left here, we have a voltage-gated calcium channel opened by depolarization. That depolarization can be due to neurotransmitter binding, hormone binding, or even stretch of the muscle itself. But in all cases, our rise in intracellular calcium is going to start to interact with this calcium-binding protein called calmodulin. And once calmodulin is activated by that calcium binding, it becomes active and helps initiate activation of a kinase, an enzyme that phosphorylates other proteins. In this case, it's a very specific kinase known as myosin light chain kinase, or MLCK. Upon activation, that myosin light chain kinase will phosphorylate our myosin. Activating it actually activates the ATPase of those myosin heads, as well as allowing it to bind to actin. You can see over here on the bottom right, our thick filaments become activated. They would then interact with our thin filaments, creating crossbridges, allowing shortening. But in this case, these are not sarcomeres. We don't have, this is an unstriated muscle, right? And so we still have thick and thin filaments. We still have shortening of the muscle cells themselves by this arrangement of thick and thin filament overlapping and the generation of tension. The thin filaments are suspended by dense bodies, shown here. The dense bodies are suspended by the cytoskeletal filaments, right, allowing this sort of coordinated cell-wide generation of tension. And keep in mind, right, a contraction is useful in many cases, but we often want to also relax that muscle after a period. And to relax it, we would have to undo the activation that already occurred, right? And that activation was a phosphorylation, right, a molecular switch, essentially. And the opposite of a kinase is a phosphatase, right? The kinase added the phosphate group. A phosphatase would be activated to remove that phosphate group and initiate relaxation of the smooth muscle.